Hi everyone, welcome to our Translational Neuroimaging educational program. I'm the moderator for this event today. My name is Ray Yang and I work at the Department of Psychiatry at University of British Columbia. Before introducing our guest speaker today, please allow me 2 minutes to have a quick introduction of our program. This educational program is nested within the lifespan working group of the Enigma Consortium, which spans more than 45 countries and involve over 4,000 researchers worldwide. Our program is inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The directors of this program are Dr. Shalala Haas from Mount Sinai, New York City, and myself, and we do have an international advisory committee. So please consider to join our mailing list if you wish to receive the notification of updates about our program. And we have a YouTube channel where you can find the recordings from previous event. I'm also recording today's event. So if you are interested, keep an eye on our YouTube channel. We will be uploading it very soon. Just a heads up, we'll only be sharing the presentation about our guest speaker, not the Q&A part. So please feel free to raise any questions for our speaker after their presentation. We are on both Twitter and Blue Sky. If you are on either platform, we would love for you to follow us. If you wish to share your works through this platform, please get in touch with us. We welcome any type of collaborations. After today's presentation, our next event will be on October 21st. Well, Yixue Feng from USC will join us to share their latest work on white matter tractography. And today, we're very excited to have Dr. Ruby Kong. Ruby is a senior research fellow at the Center for Sleep and Cognition, National University of Singapore. She completed her Ph.D. under the supervision of Dr. Thomas Yeo. Her research focused on developing machine learning algorithms to investigate individual differences in brain organization using functional MRI and examine how these differences related to human behaviors. More recently, her work has expanded to personalized brain stimulation, which is very exciting. In the next hour, Ruby will share with us the newly developed Network Correspondence Toolbox and how to use it to report novel neuroimaging findings. She will also share a multi-session hierarchical Bayesian model that generates high-quality, individualized functional brain parcellations and how this personalized brain parcellations can be used to clinical applications. So, Dr. Kong, now the time is yours. Thank you for joining us.
Thank you. Thank you for the nice introduction. I think I will share my slides first. Okay. I hope this works. Great.
Yeah.
Okay. So thanks for having me here. It's a great pleasure to join this great educational program. And for those who don't really know our work, we mainly work on deriving personalized brain parcellations. For a while ago, actually it's my supervisor Thomas, and he mainly focusing on like a group level parcellation. And he got this well-known Yo-70 networks, and so during my PhD with him, I have been focusing on how to expand these great work into like personalized parcellations. And recently, we have been developed some interest to see how this work can be translated for clinical applications. And we have several very cool clinical trials ongoing. And so I'm happy to share with you guys for what we have been doing recently. Okay, let's see. Sorry. So we mainly working on resting state functional MRI image. So the resting state functional MRI, this video is from Daniel Margulies showing how the functional MRI signals fluctuating up and down together. So this was recorded when the subject is lying in the scanner at rest without doing any specific task. So in our lab, we have been working a lot using the resting state functional image. So main reason is because at that time we don't collect any of our own data. So we work on the large scale data sets, publicly available, like a Human Connectome project, Genomics Superstruct project, and Yucca Biobank, something like that. So a lot of our work is developed based on the resting state functional MR image.
Sorry, guys. I think,
Sorry, I think,
yeah, you were frozen. Yeah, I
oh, yeah,
think this is the
I think I
network.
got kicked out somehow.
Oh,
Just
really? That's
oh
weird.
God.
Yeah,
Let
yeah.
me reshare again. I don't
Okay.
know where I stopped. So, but I don't think we miss any cool part yet. Is the sharing okay?
Yes.
Okay, okay, cool. Okay, so we work on the resting state data, and one great thing we have found for the resting state functional RMA image is that it can be used to explore the large-scale brain networks. So, for example, if we put a seed region and showing here as a black circle, and we correlate its time course with the entire brain, and we will see a nice resting state functional connectivity pattern. And if we move the seed regions around, and we see the resting state functional connectivity pattern changes a lot. So so many people have been using resting state functional connectivity to derive like the large scale brain networks. And one of the well-known like networks, as I mentioned, is like from Thomas. He's like a Yo 2011 17 networks. And after like a few years, we also derive more like a fine-grained area level pulsation. And this is the Schaefer 2018. And it has like a wide range from like a 100 RIs to like a 1000 RIs. Of course, we are not the only one in the world like working on this field. There are many great work, very cool like atlases out there also. So for example, these are the Power 2011 17 networks, and also the Co-Antisavic 2019 brain networks, which group the Glacier 3060 RIs into like 12 networks. So of course, we also notice that there are some differences. For example, in our lab, the parcellations or like brain networks are mainly developed using the data in the fSRH space. And on the other hand, the worship people, for example, the worship people, they mainly work on the data in the FSR surface space. And there are many other LSEs out there in the volumetric space. So for example, the Shen 2013 LS, the Shire 2012, and also the well-known ICA ICA spatial maps. So all these like brain atlases are out there, but they are like have different number of brain networks. They also have like they are also derived originally in different brain space. So to study these like brain atlases out there, what we do is that we think, oh, maybe we could just concatenate all these brain atlases, like brain networks together, even though they are like all derived using different algorithms and using data in different space. So we collect like a 16 most representative atlases, including our labs like a Yo 2011, a Schaefer, and my own like Parcellations and the power and the ICA maps, for example. And what we do here is that we want to see the spatial similarity across all the brain networks from these different LSEs. And then we perform a hierarchical clustering and to see if there is any correspondence across the brain networks, even though they are all developed using different methods. So what we found here is like this matrix, we group all these networks into 15 clusters. And where we can see there are nice, beautiful like a diagonal structure indicating that it looks like these networks having some like a correspondence, even though like at least spatially similar across each other using the hierarchical clustering algorithm. And if we're focusing into details of like each cluster, and we can find for like for example for the cluster 3, most of the brain networks were spatially similar, were named also like very similarly. So some networks was named as visual B, from one atlas, and then some other atlas call it like a primary visual or medial visual. However, there are also a very interesting case. Like for example, if you look at the cluster 15, even though all these brain networks are spatially very similar, the name is very different. So one of the atlas call it the salience network, and the other atlas will call it like a control C network. So this has been pretty annoying, like even for our own case, because when we talk about like the spatial similarity, we usually assume like different people call it like similarly, but this is not true. And what makes things even more problematic is that sometimes a network can be given the same name, but it has very different spatial topographies. So what I've shown here are the salience networks from 4 different brain atlases. So we can see that although like all these like brain networks are called salience network, but they are all very different. So we ourselves have been through a lot, like because we also got confused by the names. And later in the when I talk about the clinical applications, you will have a much better understanding of like why this is so problematic. So because of these troubles, we developed the Network Correspondence Toolbox, NCT, to help people to examine and report the spatial correspondence between the neuroimaging results and all these like different kind of brain atlases. So the next few slides, I will explain like a few usage scenario of how you can use the NCT toolbox. And I will also provide a few like examples of like so that you have a better sense of why this toolbox is useful. So one of the usage scenario of the NCT toolbox is that it can be used to explore the network correspondence between 2 different atlases. So in this example, the reference atlas is the Yo17 networks. And as I mentioned, it was derived using the fSRG6. And then, so say if we have a new atlas, which is the Golden 17 network in this case, and we want to compare the network correspondence, we'll see like one is derived in fSRG6 and the other was derived in FSR space. So the toolbox will first project the Golden 17 networks from the FSL surface space to the fSRG6 space to be the same as the reference atlas. And then the toolbox will then perform the spatial similarity and then also a significant test to test whether the 2 networks are, like any 2 pair of the networks are significantly similar. As a result, the toolbox will provide a very sufficient network correspondence summary. So the first summary will be visualized as an overlap heat map, where you can see is a matrix where the left side shows the reference atlases, and on the top row shows the networks from the other atlas. And in this case, since even though the Yo17 networks and also the Gordon atlas were derived using different algorithms, but we can see they have a very nice diagonal structure. This means that even though the number of networks are also different, but there is a nice spatial similarity between the networks. And the toolbox will also provide a summary table. And in the summary table, we provide exact values for the spatial overlap and also the p-values. So another more widely used case is that to use the NCT toolbox to study the network correspondence between any user-defined data and the existing atlases. So here I'm gonna use the HCP 2-back to 0-back task contrast maps as the example. So the input data is already binarized in this case. So together with the input data, the user also need to provide a config file. The config file is pretty simple. You just need to contain the most basic information, such as you need to give a name, and then you need to mention like what is the data space, and also whether the data is binarized or not. And you also can like provide the atlas list. If you don't, like we will just compare with all existing atlases. So the atlas list can be a list of the atlas you are most interested. So in this case, I use 4 different atlases as examples. So the toolbox will first project all the atlases included in the atlas list to be in the same space as the input data. And similarly, it will also compute the spatial similarity and also perform the statistical test to get to generate a network correspondence summary. So as a result, since now we have in this example, we have 4 different LSCs. So therefore, we will visualize the results as a network clock. So here, only the brain networks are significantly overlapped with the input data will be shown here. And the larger font size indicates that this network has a much larger spatial overlap with the input data. So in this example, we can see that so this input data is overlapped with some so-called frontal parietal network, and sometimes it's called like a control A or control B networks. We also provide a network radar map for each LS. So the network radar map is supposed to give a more detailed spatial overlap values for all the networks within each LS. And similarly, we also have a summary table with the p-values and the exact dice values. So next, I will give 2 examples of how we use the NCT Toolbox to explore the network correspondence with the UCL Biobank ICA maps. So for example, here we show, I think I forgot to change the name. It's supposed to be, I think it's ICA component 3 instead of 5. So we can see when we have this ICA spatial map as the input data, and we can find that it has a very obvious visual network pattern. So not surprisingly that this input data will overlap the most with the visual networks from different LSCs. However, when the ICA map has much more complicated structures, so for example, in this case, and we'll find that actually if the input data is the ICA component 5, and the some network will call it like a singular percolor network, and some LS will call it dorsal attention or like a salient ventral attention network. There is no general name to match with this input data. So this is very troublesome. That's why we suggest that the user is supposed to provide a complete like a summary table with the overlap values and also the p-values if they want to justify, if they find it very troublesome to justify whether the input data is corresponding to like any specific network. So the toolbox is now available in the Python index package, and it's supposed to be very easy to install just using the pip install CPK network correspondence. Of course, I have received a lot of requests from different users either to upload the new their own LSCs to the toolbox, which is I'm very happy to. And actually, I already have a long waiting list. It's the only problem is because I'm the only one maintaining the this toolbox, so everything has been a bit slow, and I work on very big major release, and I have promised to everybody write me that I will release soon, and I'm really sorry that I haven't pushed it faster enough. Anyway, but guaranteed I will do a major release in the next few months. And because I want to make things much easier, currently it's a bit slow because the toolbox containing all the atlases from different people, and therefore it has a huge data downloading part. And I'm currently optimizing the toolbox to see if there is any way I can speed up the thing. I'm happy to collaborate if anyone has want to help me with that also. So here is the GitHub page, and you can post issues and feel free to download and feel free to write me. I may be slow, but I'm quite responsive. So far, I've been spending a lot of time to discussing about the group level parcellations. So all these currently open atlases are derived using group average data. However, my own interest is actually to derive the personalized parcellations. So because we know that the group level networks do not capture enough individual specific informations. So I have developed algorithm to derive so that it can derive a high quality individual parcellations using limited scan duration. And we showed that these parcellations can be used for behavior prediction, and it can predict the cognition, emotion, and personality pretty well. So here is an overview of how this algorithm structured. So we call it multi-session hierarchical Bayesian model. I won't go into details of like how this mathematically how this works. So basically, at the top level, we try to model the inter-subject variability. And after that, we try to model the inter-session variability because we know that even it's the same person, like across different days or like even like across hours, it may have certain different like a functional activity variability. So at the second level, we try to model the intersession or like a within subjects variability. And after that, we model the interregion variability. So this is the variability across the brain regions within each network. And then we have all these priors, and we have a pre-trained model where these priors were estimated using ACP data and GSP data, I think. Also, so we learned these priors and constructed a pre-trained model so that user can just use the pre-trained model to apply on their single subject, maybe only with like limited scan duration. For example, in this case, in our own case, we have been explore like a usage scenario where user only have like 8 minutes, and the shortest one we have tried is the UK Biobank, which is only 5 minutes. And then we can derive a nice personalized individual brain networks using just like, for example, 5 to 10 minutes of data of the unseen test subject. So we use this MSHBM model to derive the parcellations for HCP data, for example. And here I'm showing you 4 representative ACP participants. And on top row, I'm showing you the brain networks derived using the resting state data from day 1 from the ACP data. And then on the second row, I'm showing you the brain networks derived using resting state data from day 2. So we estimate the brain parcellations separately using day 1 and day 2 so that we can see whether there are any like individual specific features that are highly replicable within individuals and are also distinguishable enough across the individuals. And here the arrow is supposed to point to you some like unique individual specific topographic features that are highly replicable within individuals, but also very different across individuals. So if, for example, if you look at the blue arrow and it's pointed actually to this tiny yellow patch, and same as the yellow arrow. The yellow arrow, it was pointed to this like very tiny yellow patch from the default network, and where we can see that for some individuals it's shown here, but for some individuals it's disappeared. And then we find this is be very interesting. However, we don't know whether these are like a biologically meaningful or not. So what we do is that, so before that, we also show that the networks derived using our MSHBM model, using just like, as I mentioned, 10 minutes of data, it can generate comparable quality to other like 2 other approaches using 15 minutes of data. And then we try to see whether these like networks are useful, like meaningful. So what we do is that we assume that if 2 subjects, they have very similar network topography, and we assume they also have similar behavioral phenotypes. So what we do here is like we then use this network topography to do the behavior prediction. And we also try on the network size where that, for example, if 2 participants have very similar like a network size, and then we assume that they also have similar like a behavior phenotypes. So using this assumption, we do the behavior prediction, and we find that actually the network topography can perform much better than the network size in terms of the behavior prediction. I mean, in the paper, we also show like a functional connectivity, of course, but I think so far we are focusing on here I'm mainly focusing on the network topographic features. So I didn't include that here. And after that, because the previous model was built purely on the cortical, the brain areas. So what we want to see is that oh, what if we can use it for deriving the individual specific cerebellar networks? So my colleague Alvin then use our cortical network, our personalized cortical networks, and then she focusing on the cerebellum. And we have two densely sampled participants from Randy Buckner's lab, where like each participant has like 31 sessions of resting state data. So these are like densely sampled subjects. So the quality of the cerebellum areas are much better than the usual case, of course. So where we can see that using our MSHBM model, for these two participants, we can derive very like a beautiful cerebellar parcellations. However, like just as I mentioned, if you are using like some data with much shorter scan duration, the cerebellar networks can be much more noisier. So we then like so as I mentioned for the MSHBM model, we try to model the inter and intra-subject variability. And however, one biggest concern is always that how confident are we that the MSHBM model is actually generalizable to a new populations. As I mentioned, the pre-trained model is trained using ACP data or the GSP data, but we are a bit unsure that whether it can generalize to another like a population derived using different acquisition protocol and also like from different scanner. So our student Shreya, and she's mainly focusing on like whether we can improve the model by considering the between population variability. So of course, like we believe the ceiling level performance of the MSHBM model is to just retrain the model using the training set from this new population and then apply to the test set from the same population. And we believe this is the ceiling performance. Of course, this is not applicable for most general cases because like especially if we are working on the densely sampled population, we don't have that many subjects to do the training, right? So here like we use the ceiling MSHBM as the baseline comparison or like a ceiling comparison. So Sharia developed two different ways to model the between population variability. The first way, we call it MSHBM-MATI, where we just retrain the MSHBM model with training sets from like 13 diverse data sets. So it's like a multi data sets training. And there is also an MSHBM++, like a much fancier version, like iPhone Pro Max things. So the MSHBM++ supposed to model the between population variability as a additional hierarchical layer. So it's like she remodeled the entire like MSHBM structure and try to consider the between dataset variability. So in my first impression, I was like, oh, the MSHBM++ is supposed to perform much better than the MSHBM-MATI. And however, this is not the case. So in our experiment, we find that actually the MSHBM++ perform exactly like the same level as the MSHBM-MATI. So in the following slides, I won't show the results of the MSHBM-MATI, MSHBM++. So as I mentioned, because we try to use like, the multiple different data sets to retrain the model, and so we consider like 13 different data sets, including the ABCD from different sites, the ENK data sets, the GSP data set, the HBN, ACP aging, ACP development, and ACP healthy young adults, and also a local data set called Singer. So these data sets are all healthy participants, but across a very wide range of the age populations. And we try to evaluate the quality of the brain networks using the homogeneity matrix. So the homogeneity matrix is basically the correlation between the time series within each network. So the higher homogeneity we think is indicates better quality of the brain networks. Of course, in the like we also perform many other like validation metrics, including using the task data, but I won't really like cover too much details here. So the conclusion is similar. So just to reassure you. So here is the resting state homogeneity metric. And what we find is surprisingly the MSHBM-MATI actually can achieve similar level of the performance compared with MSG and ceiling. So, and both the MSG and ceiling and MSG and multi performs better than the group average methods. However, we didn't just stop there, because as I mentioned, we recently have developed interest to translate our research angle to more like clinical applications. So one interesting question is that whether the MSHM model can generalize well to the neuropsychiatric cohorts. So what we do here is that we including like first we include the three local data sets with the we only consider the APIC and CHIMES and NGCG. These are the local depression and schizophrenia data. And we also including the 1,024 psychiatric patients from the TCP datasets. And then what we do here is that for each of the datasets, we want to see the performance across these three approaches. And we find that similarly even though the Amsterdam multi is trained using certain healthy populations, but it looks like it can achieve the ceiling level performance with the Amsterdam ceiling, even in the neuropsychiatric cohorts. So we think, oh, that's really cool, because recent work from WashU, Charles Lynch, he has a very, very inspiring Nature paper where he looked into the salient, like their salience network in the depression patients. So that's why we think the Institute work is very useful, because when we look into this paper, we saw their salience network is our salience network, but interestingly, it's not. And it confused me for a while. And so yeah, be very careful with the network naming comment, especially if you are dealing with like a brain analysis from different lab. And this is very tricky. Okay, so in this work, Chuck find that the salience network expanded like nearly twofold in the cortex of the highly sampled depression patients. So here is the figure from still from his paper, where in figure A, we see the salience network, their salience network in the healthy controls. And in figure B are the salience network, the personalized salience network in 3 densely sampled participants. Where we can see that clearly that in these 3 depression patients, this network has much bigger network size compared with healthy controls. We think this is very cool because we have our own personalized like a parcellation algorithms, right? So we're like, oh, whether we can replicate that in our own data sets. So we use the Institute of Wealth, and we find that actually the salience network corresponding to our control C network. And we look into the exact same metric, and we find that in our own data sets, we also find that our like depression patients can achieve also like a much bigger network size in our control C network compared with the healthy controls. So we are like, oh, I think like it's great like when you are able to replicate some other people's work, and especially like this is very different algorithms and also very different populations. So and then we think, okay, it looks very interesting to just expand it further to the depression, and we also like be confident that we can do in the depression patients. So what we do is like we then want to see whether we are able to using these personalized brain networks for personalized brain stimulation for depression patients. So for those, I don't know like the people's background who joined this education program. So here is just a short video of me receiving that's me before the clinical trial. I'm aged a lot after the trial, unfortunately. So this is this is the trouble part when you're starting to do your first clinical trial. You just grow by hair. And then in this video, I will receive the TMS pause, where our clinician try to stimulate my hand and my foot. And this is using a purely like a manual method. This is the what we call the transcranial magnetic stimulation. I think Ruiyang is an expert on this field. Okay.
Then I'm gonna make you right hand switch.
I'm not sure you're able to hear the sound or not, actually.
Yes, we can.
Oh, that's great. I see my left hand is twitching.
Wait, but just now the right hand actually the left hand twitchable.
Sorry, Thomas is holding the camera, so we'll see his voice from the background.
Then make your, now you make your left hand move. Right hand move. And now you notice that this is actually not that floating. I'm gonna make a almost on the wall. Make your right foot, left foot, sorry. Oh, now I'm gonna make your right foot. There we go. Okay. How you?
Okay, so this is me, my very first experience with the TMS stimulation, and you can see that when the clinician try to stimulate my hand, and especially my left and right hand, my left and right foot, he has to iterate a lot, like through different like spot, so that he can find the thing. But the thing is, like, our clinician is a very good like a clinician, so he has been doing this for many years, and even for him, it's not that easy to just like to find the very correct spot at the like just a few try, right? So this is he's also using a very different coil, and you will see actually it's not so visible in. Oh, I think maybe it's visible. It's an angle coil, so that will cover like a much wider range of the brain areas. So and he's also using a much stronger dosage, so that can stimulate my the correct spot. So this is the transcranial magnetic stimulation. For those who don't know the mechanism, basically it's just the coil will generate a magnetic field, and when the magnetic field passing through the brain tissues, it will generate very tiny like a electrical current, and the E field will cover through like different range of the cortex areas, and if the thing is like the TMS cannot reach very deep into the skull, because the magnetic field is has a limited like the gauge. So that's why for when people use the transcranial magnetic stimulation for the real clinical application for depression patients, they have been facing troubles. So one of the traditional way to use the TMS for the depression patients is that using this so-called the beam X-ray approach, where in this figure you can see that the patient will wear a white cotton like a cap, and then the nurse will draw the several landmarks in the cap, and then they will take the measures of like the size of the skull and things like that. So this is a pure scale-based measurements, and it's we think why TMS we could achieve much better efficacy for TMS is because we think the BM3 approach is not so reliable. So back to the 2012 and 2013, Michael Fox has proposed this great idea of that. What if we can use the functional connectivity to guide the TMS target? So basically, the idea is that we believe that for the case of depression patients, we should stimulate the area in the dorsolateral prefrontal cortex that most anti-correlated with the subgenuine cingulate cortex. So on the left figure shows the group level SGACC-RI. On the right shows the group average functional connectivity of the subgenuine cingulate cortex. So here the black circle indicates the area most anti-correlated with the highest anti-correlations, and so it will be the target spot. And in this paper, Fox also mentioned that there's nothing stop us to like move from the group average to the personalized functional connectivity. So therefore, after that, many paper has been looking into whether we can use the personalized functional connectivity to derive the TMS target for depression patients. So one of the very cool work is from the Robin Cash and Andrew Zaleski in 2021, where they have a bunch of the TMS, a bunch of patients have received the TMS treatment. So they know the actual target spot, which is using I think in this paper is using 5 cm rule. I could be wrong. And then it's a scalp-based measures for the actual treatment target. And then they're also deriving the optimal target using the resting state functional connectivity, using their own algorithm. So the hypothesis is that if the optimal target happen to be very close to the actual target, then therefore they expect this particular patient to respond very well. However, if the optimal target is very far away from the actual target for another subject, then they expect the treatment outcome to be terrible for this particular subject. So if you draw this correlation as a scatter plot, they expect there is a negative correlation between the clinical improvement and the distance to the optimal site. So they look into their data and then they find they are able to derive this very beautiful negative correlation. So this is very cool, is because the finding is able to be replicated by Sean Sticky using a very different data set, and also they are using a very different personalized FC guided TMS target algorithm. So therefore, so here we can see that similarly they have the actual TMS size. I think this one is using BM3, and then the personalized TMS target using a different algorithm, and then we can see in figure B there is a very beautiful negative correlation. So we were also like inspired by these previous works, and we are like, so we could it seems like we could also using the personalized brain networks for TMS target. So what we are doing is that we use the MSHBM model to first derive the personalized target, to first derive the personalized brain networks. And then we also consider the individual cortical folding pattern shown here, because as I mentioned, the TMS, the TMS magnetic field couldn't reach very deep into the brain. It's usually like a 3 cm around. So what we are doing is that we use the individual cortical folding pattern and we restrict the target spot to be within the superficial areas for the cerebral cortex. And then we have a hypothesis that we believe the target spot supposed to be situated within the attention networks. So in our case, we have the salience attention networks, which are these two pink and purple color networks, and we also include the two dorsal attention networks, because for some subjects they will have dorsal attention networks also appear in the DLPFC areas. So for example, you can see a tiny green bit here. And then we compute the functional connectivity with the subgenuine cingulate cortex, and we develop the threshold-free tree-based localization approach to find the target spot that are most anti-correlated with the SGACC. So the key part is that, as I mentioned, that because we want to restrict the cortical fold, restrict the TMS target to be situated near the skull. Therefore, there are several like a threshold we need to tune. And of course, in the real clinical case, we cannot rely on the clinician to like manually tune it every time for each different single subject, right? So that's why we came up with this algorithm that we can do everything free. So in our clinical trial, we don't need to tune anything, and we have tried on different data sets, and this algorithm has been worked greatly. So as I mentioned, then after we think we have finalized the TMS target algorithm, we're like, okay, we're ready for the open label trial. So we launched our first open label trial, and it's called APIC TMS. And Alice is actually our very first patient, and she was referred to our clinician by her own psychiatrist. So she diagnosed with depression when she was only 20, and she has committed suicide. She dropped out from the top university program also, and she tried like a different antidepressants, and unfortunately, like nothing really works for her. And she actually also tried the so-called ECT, and basically the ECT will is actually deliver the direct current into the brain, and it will induce seizure. However, Alice have very severe side effects after the ECT very first session, so she has to stop. So she also tried the standard TMS where we use the BM3, where our clinician used to use the BM3 approach to derive the target spot. And also, unfortunately, it also doesn't really work for her. So when Alice arrived, we scan her at our center. And in our clinical trial, we collect 2 runs of the resting state data. Each run is 10 minutes. And we use the resting, so these are all collected like 1 week at most before the real treatment. So I think we have a few patients that were scanned like a few weeks earlier than the treatment week. But this all supposed to be collected within a very short range from the treatment day. And then we use our own algorithm to derive the stimulation target location for Alice. And for our open label trial, we follow the same protocol as the so-called like a SAINT protocol, where the patient will receive a 5-day TMS treatment and is 10 sessions per day. And each session is a 10 minutes ITBS. So basically every patient has to come to us a very early morning, and every hour they will just receive a 10-minute session TMS treatment and with a 50 minutes break. And then we do the next 10 minutes. So it's very intense. However, we can see that Alice enjoyed a great response to our TMS treatment, and compared with previously, she has tried the standard TMS, but unfortunately it doesn't really work for her. So in our open label trial, we have finished collecting 20 major depression patients, and that's the sample size. Unfortunately, it's the open label trial. So we are now collecting more data, but for the open label trial, it only has 20 depression patients. Or one thing I have to note is that for our case, because we want to test the effects for real world patients. So in this case, these 20 depression patients all have very different comorbids, including anxiety, OCD, and etc. So we have total about 9 different comorbids, and some subjects may have more than 3. I think we have a patient they have 5 different comorbids. So here is the distribution of the final target spot for these 20 patients, and we overlaid everybody's target together on top of a group average brain network, and we can see that so that we have a sense of how this target distributed across the cortical areas. So where we can see that most of the target spots are situated within the salience. These pink salience attention networks, even for the group level case. However, there are certain subjects whose target spots actually is situated in the different network in the group level. This is because the group level, as I mentioned before, does not capture enough individual specific topographic features. And here are 4 representative patients where we can see so the green bars represents the target spots, and we can see that they are all nicely situated near scalp. So we also calculated the distance between the target spots between each pair of the patients, and so we can see that actually the maximum distance between the target spots for each pair for a pair of the patients can be up to 2 cm. And we also interested to see how different the functional connectivity guided TMS targets compare with the BMP3 derived targets. So for each patient, we calculate the distance between our personalized target and also the BMP3 target. And we can see that actually the maximum distance can be up to 3 cm. So we have finished the treatment for these 20 patients, and we can see that overall we are able to achieve a 70% response rate. So in the exact same population in the same center, we used to only be able to derive a 21% response rate. So our clinician is very happy that, and we're also very happy we are able to help these patients. So here is a very short video of our PI when he first received the theta burst TMS, As you know that the previous video, when you see me receive the TMS treatment with the TMS pause to simulate my hand movement and my leg movement, these are just like single pause. The ITBS is much like intense. So
something like this 30% very low. I didn't even feel it.
So it's a very painful experience actually. And this is actually very low dosage. It's only 30. So it's either Thomas being very sensitive, like or it's because we have very good patients because most of our patients able to finish the trial. So as mentioned, we also have several trials going. We are currently collecting data for our randomized control trials. So in the randomized control trials, Spark, where we have 2 arms and 1 arm, the patients will receive the TMS target using our personalized functional connectivity guided approach. And another arm will receive the TMS treatment using the Beam F3 approach. We aim to collect like 70 local major depression patients. Currently, we only finished 20-ish, I think. So still a long way to go. And besides that, there's nothing stopping us to move to like other mental health disorders. So you can tell that actually the algorithm itself is not very restricted to the depression patients. So we also derived the personalized, for example, the personalized anxiety target, which is shown in this figure. So in summary, I have mentioned that we developed a toolbox. It's called like a Network Correspondence Toolbox. We hope this toolbox can help user to like better examine and also report the spatial correspondence between their neuroimaging results and also the functional brain atlases they're interested. And then I also briefly mentioned we have algorithms called like a multi-session hierarchical Bayesian model where we use it to derive high quality personalized brain networks. And in the initial version, we consider only the inter-subject and intra-subject variability. And we showed that the size and topography of these personalized brain networks actually can be used for behavior prediction. And we have further improved the multi-session hierarchical model to be like a plus version. So where we use the multi-data sets to train this model, and we find that the multi-data sets trained model is able to achieve ceiling level performance in the new healthy controls and also the psychiatric cohort. And lastly, I mentioned our clinical trial where we find that actually we can use the brain stimulation. So we can use the brain personalized brain networks for brain stimulation approaches. And we find that we can actually also derive like reliable personalized targets. I would like to thank our funding support and also our lab members, my collaborators. So thank you. Here is the code and for the parcellation approaches where you can use here. And also because this is a education program, and here are the exact locations where you can find the brain network codes. And so actually I have 2 different brain parcellation approach to derive the 2 different personalized brain parcellation approach. The first one is for the brain networks, and the second one is for for more fine-grained area level parcellations. So both are currently available already. And here is a very short I wouldn't say introduction, like a very short overview of how you can use this pre-trained model. Just remember that for the currently, we only released the pre-trained model for a single data set, which is either the ACP data set or the GSP data set. In the near future, we will, I think Shreya will release a much fancier version using the multi-data set trained model. So to use the model, here is the script, which is actually coded in MATLAB. It's supposed to be very easy to use, just like one single wrapper with a bunch of input parameters, including like the list of the MRI data, and if you have like a sensoring and what is the sensory list and what is the data space you're working on and things like that. So it's supposed to be very easy to use. And also like feel free to ping me if you encounter any problems. We have a lab. If you go to our lab repo, and you will also find we have a I think it's Gmail group or something where you can post any of the problem you have encountered so far. So our people are usually very responsive. And I also want to highlight one thing is because all our parcellation approaches were derived using surface data. Therefore, the resulting parcellations naturally will be in the surface space. However, since like when we do brain stimulation or like when we work on some data for like a epilepsy case, for example, and it requires us to work on data in the volumetric space, in the native volumetric space. So just to briefly mention that there is a way like I can just project. Also, actually, we always do that. Is like we can project the individual parcellations from the original surface space to the native like a surface or native volume. Yeah, here is just an illustration of how it will look like if you do these projections. Yeah, that's all. Thank you. Any questions?
Yeah. We have 3 minutes for maybe for 1 or 2 questions. Thank you, Dr. Kong. Very wonderful talk, very impressive work.
Thank you.
Yeah.